Go ahead.
All right, can everyone hear me?
Yes.
Yes, we can.
Okay, right, so as you said, our research was on iron oxides, and that might bring up a question. What are iron oxides? So iron oxides are actually found all over the earth. You can find them in soil and all different types of rock formations. The project we'll be talking about today focused on the more fundamental side of research, where we started to analyze some different properties and characteristics of these materials. So we like to call this our garden project, and it's the start of a 5-year process. So as the title suggests, this project had to do with the outdoor garden and high tunnel on Concordia's campus, and I'll talk a little more about that in the next slide. But during this project, we worked with Kyle, Anna, and Gabrielle, who helped us with the preparation of different plant beds that we're using for our research. So I wanted to say thank you to them right away. Now, as for the goals of this project, we didn't begin this research with a specific goal in mind. More so, our goals were to explore fundamental environmental magnetism. We did have some ideas as to what we're going to look for, though, namely how different environmental conditions would affect phase conversions in our iron oxides. And to do this, I'll give a brief overview of our project. So basically what we did is we mixed 5 different iron oxides into plant beds that would be subjected to different conditions, primarily temperature and moisture differences. So one set of plant beds is in the outdoor garden, where there's going to be rain and snow, and in the winter there'll be temperatures down to -40 degrees, and in the summer up to 100 degrees. And samples from these beds are going to be compared with those in the high tunnel, which is like a sort of greenhouse. And there's going to be a more temperate climate where the temperature doesn't get below freezing. There's a regulated water system. And our hope is that over time, as we collect samples from these different sets of plant beds, we'll be able to come to a conclusion about whether the environment actually has a significant impact on the phase conversions in iron oxides.
All right. Now I'll talk a little bit about the different iron oxide samples that we use and how the factors introduced on the previous slide can be seen in their transformations. In the photo from left to right, we have 5 different samples. They are Quadtite, a sample that we termed XFEO, which is a mix of magnetic iron oxide phases, Hematite, Megamite, and Magnetite. The difference in these phases is either that they are chemically different, say with different compounds of oxygen, iron, and hydrogen, and might use different oxidation states of iron, such as iron 2 or iron 3. Or if they are chemically identical, they are structurally different. You may never have heard these materials before, but they can be found in your daily life. For instance, the top 2 are found in technology and are used in hard drives, so they can be found in, say, cell phones and laptops. And then the bottom phases you might better know as rust. First, let's go through some of these in depth. First, we have Quadtite, which occurs in sedimentary rocks and sediments naturally. It has a hexagonal crystalline structure. It is quite thermodynamically stable. It only makes one conversion, and that's to one of our other phases, hematite, at around 300 degrees Celsius. It is an antiferromagnetic material. It has a characteristic Néel temperature, or the temperature below which it exhibits antiferromagnetic behavior, of 120 degrees Celsius. And it has a hyperfine field, which is related to a type of spectroscopy that we did on our samples, which we'll talk about a little bit later, of 38 Tesla. And just a fun fact, it is named after a German poet and scientist. Next, we have magnetite. Magnetite is interesting because it contains both iron 2 and iron 3 in its structure. It is responsible for the magnetic properties of rocks and is formed in various organisms such as bacteria, or you can also find it in the beaks of pigeons. It holds a large interest in a variety of disciplines, including technological, as we talked about previously, and medical, where it's currently being used to help treat cancer and as a drug delivery system. Magnetite is ferrimagnetic, which gives it a Curie temperature, or a point at which below it has a permanent magnetism of 580 degrees Celsius. And because it contains both iron 2 and iron 3, it has two magnetic hyperfine fields at 46 Tesla and 49 Tesla.
Right, so next we have hematite, and this is also found in sedimentary rocks and soils. And as you can see, it has a very bright red color, so it's actually common in pigments, and it's also an abundant phase of iron oxide on the surface of Mars, hence the name red planet is where it gets its color from. It has a similar hexagonal structure to goethite, and it's also antiferromagnetic. It has a Néel temperature of 675 degrees Celsius, a hyperfine field of 51.8 Tesla, and it's also extremely stable. It's the end phase in transitions. So whenever an iron oxide is going through its different phase conversions, if it goes all the way through, it's going to always end up at hematite. Now for Megamite, it has an isometric structure to magnetite, and it occurs in soil as an oxidation product of magnetite. It's actually an intermediate phase between magnetite and hematite, so it doesn't have a hyperfine field or Curie temperature, but even still, it's another important magnetic material in technological industries, and it's very magnetic.
All right, so I'll talk a little bit about the preparation and how we started this research project. For our garden project, we first needed to prepare a research area. And to do this, we mixed our 5 different samples shown a couple slides ago into 10 different soil beds, 5 inside the high tunnel and 5 in an outdoor garden. The picture on the right is a really nice photo of all of our samples laid out, so you can see their vibrant colors before we mix them into the soil. And now here are some of the schematics of the work we did in the garden. On the bottom right, you can see a photo of Concordia's high tunnel. And above that is a diagram of what is below the surface of the high tunnel. So essentially, to extend the growing season, the high tunnel has a layer of sand and heated air pipes powered by the solar panel seen in the photo that circulate warm air in the winter months. And then beneath that is a layer of gravel to help drain excess moisture. Additionally, the plastic, which is currently up in the photo you see, can be raised or lowered depending on weather conditions. And then in the diagrams on the right, we have different layouts of how we mixed in our soils. Again, the colors correspond to the different samples we used, which can be seen in the caption on the right. And now just to highlight some of the environmental variables that we're looking at, we have a photo of the high tunnel in the summer compared to one taken this winter. As you can see in the winter, the ground is covered in snow, so we have different moisture conditions in the outdoor garden and the high tunnel where it is completely covered in plastic, so there's no snow cover. Additionally, as we said, the heated air pipes in the high tunnel keep the ground from freezing. It stays around 36 degrees Fahrenheit. And then other than temperature and moisture content, we encourage those in charge of the garden to use the beds with our samples normally because we are mainly interested in these environmental factors. And just a little bit about our sample collection. So after we mixed our samples into the ground, we let them set for a couple weeks to settle. And then we took core samples and divided them up based on depth. Those have been left out to dry and are currently being run through Mosfet spectroscopy to kind of get a background reading. And we'll talk a little bit about what Mosfet spectroscopy is next.
Okay, yeah, so the majority of our analysis comes from a process called Mosfet spectroscopy. So as you can see on the graph on the right, we have open circles that represent the experimental data points. We also have a red line that's the line of best fit, which helps us see different deviations. You can also see a blue line that shows the Fe3 contribution to the sample and a magenta line that shows the Fe2 contribution. And pretty much this just tells us what the different oxidation states of iron are present in the sample. So here we have 13% Fe2, 87% Fe3. And the reason this is useful is because it tells us the composition of the sample, which can then tell us more about the fundamental properties of these materials and how they'll interact with each other. Now, continuing on with that same logic, you see another similar graph. However, here there are 5 different phases of iron oxide shown. So to a trained eye, you can actually just look at these peaks and tell why and how that these phases are different. Things you're going to look for are the hyperfine fields, isomer shifts, and quadrupole splitting. Now, for our final analytic technique, we used something called a hysteresis loop. And this is just another technique that we use to identify magnetic materials. So shown here is a sample that shows predominantly paramagnetic behavior with some minor ferromagnetic contributions.
About 2 minutes left.
Yep, perfect, because that's actually all we have left. So thank you. Yeah, and any questions?
Any questions for them?
So going right back to the beginning, you mix these things into a garden. I thought you were going to start talking about tomatoes and stuff. Did things grow in the garden? And if so, did your stuff impact how things grew? Or was that not the point of the project?
Yeah, so I think we tried to have the people who were taking care of the gardens just use our beds as they would a normal bed, just so we could get the full environmental impact of these different conditions. I'm not 100% sure.
You did not analyze any of the plants that were grown?
No,
okay,
we did not.
No, our interest it's not how the iron oxides you change it with the plants, the microorganisms, with the weather. So we are not interested in the plants.
Where are you going next with this project?
Well, we know that.
Thelma, let the students answer the question.
Okay, well, basically, I think we're planning on taking samples once a year of each individual plant bed, and we're going to be able to look at the dirt samples that we take up, and then we can use more Mosfet spectroscopy, hysteresis loops, in order to see how and if these iron oxides actually have converted phases.
Any further questions? All right, let's thank our speakers. All right, and our